GLUT transport system. GLUT transporters are also known as glucose transporter are the family of protein that facilitates the transport of glucose across cell membrane. They play a crucial role in maintaining glucose homeostasis and supplying cell with the necessary energy source. Today we will give you an overview of GLUT transporter structure and classification, tissue distribution and localization, key, key GLUT isoform, glucose transport mechanism, regulation of GLUT transporters, and clinical relevance. GLUT transporters are integral membrane protein with 12 membrane spinning domain. They are categorized into different isoforms based on their amino acid sequence and tissue distribution. The human genome encodes 14 members of the GLUT family, designated as GLUT1 to GLUT14. Different GLUT isoforms are expressed in various tissues and cell types. GLUT1, 2, 3, and 4 are among the most well studied isoforms. The expression and localization of GLUT isoform vary depending on the tissue's energy demand and glucose utilization. GLUT1 widely expressed in many tissues and involved in basal glucose uptake, it is particularly important for glucose transport across the blood-brain barrier. GLUT2 found in the liver, pancreatic beta cells, and the basolateral membrane of enterocytes. It plays a role in glucose sensing, regulation of insulin secretion, and hepatic glucose uptake. GLUT3 highly expressed in neurons and facilitates glucose transport into the brain. It has a high affinity for glucose, ensuring efficient uptake even when glucose levels are low. GLUT4 primarily found in adipose tissue and skeletal muscle. It is insulin responsive and play a major role in insulin stimulated glucose uptake into these tissues. GLUT5 primarily responsible for the transport of fructose rather than glucose. It is found in tissues such as small intestine, testes, and kidneys. It plays a role in the absorption of dietary fructose in the small intestine and the reabsorption of filtered fructose in the kidney. GLUT6 exhibit a high sequence similarity to GLUT1. It is predominantly expressed in liver, spleen, and kidney. Its exact function and physiological roles are still being investigated. GLUT7 found in a small intestine, colon, and testes. Its function and regulation are also not fully understood. GLUT8 expressed in various tissues including adipose tissues, skeletal muscle, and testes. It plays a role in glucose uptake in insulin sensitive tissues. It also transports fructose and may contribute to fructose metabolism. GLUT9, known as a urate transporter, involved in the transport of uric acid, is found in the liver and kidney, where it plays a role in urate reabsorption and homeostasis. GLUT9 isoform also have some glucose transport activity. Glucose transport mechanism. The GLUT system is like a transportation system that helps glucose move in and out of cell. It uses GLUT transporters as doors, allowing glucose to enter the cell easily when it's needed. The system is regulated to make sure glucose transport works efficiently. Once inside the cell, glucose gets turned into energy to power the cell's activity. Step 1. Glucose concentration difference. Glucose is present in higher amount outside the cell compared to inside the cell. This difference in glucose levels create a push for glucose to enter the cell. Step 2. GLUT transporters. GLUT proteins are like doorways on the cell surface that allow glucose to enter or exit the cell. They act as gatekeepers controlling the movement of glucose in and out of cell. The next step involves binding and entry. When glucose is nearby, it attaches to the GLUT transporter like a key fitting into a lock. This binding triggers the GLUT transporter to change shape and open a pathway for glucose to move inside the cell. Movement inside the cell. Once the pathway is open, glucose easily slides into the cell because there is a high concentration outside. It's like going down a hill. Glucose naturally moves from where there is more of it to where there is less. Efficient and easy. The GLUT transporter makes glucose transport easy and efficient. It doesn't require the cell to use any extra energy. It's like a free ride for glucose into the cell. Regulation. The activity of GLUT transporters can be controlled by different signals in the body. For example, insulin is a hormone that can increase the number of GLUT transporters on the cell surface, allowing more glucose to enter. Once inside the cell, glucose gets processed and used as a source of energy and goes through different chemical reactions to produce energy that the cell needs to function. Regulation of GLUT transporters. GLUT expression and activity can be regulated by various factors. Insulin is a key regulator of GLUT4 translocation to the plasma membrane, enhancing glucose uptake in insulin responsive tissues. Hormones such as glucagon and epinephrine can also influence GLUT expression and activity in specific tissues. How insulin regulates GLUT? Insulin is produced by the pancreas and is released into the bloodstream in response to elevated blood glucose levels, such as after a meal. When insulin is released, it binds to specific receptors on the surface of target cells. This interaction triggers a signaling cascade inside the cell, leading to the activity of a protein called AKT. AKT in turn phosphorylates to another protein called AS160. AS160 acts as a brake for GLUTs. By phosphorylating AS160, AKT effectively releases the brake, allowing GLUT to move to the cell membrane. When the GLUTs are present on the cell membrane, they can facilitate the transport of glucose from extracellular fluid into the cell. In the absence of insulin or when insulin levels are low, AS160 remain active and prevent the GLUTs from reaching cell membrane. This regulation ensures that glucose uptake is reduced during fasting or times when blood glucose levels are low. Clinical relevance. GLUT mutation. Mutation in GLUT genes can lead to various glucose transport disorders, including GLUT1 deficiency syndrome and Fanconi-Bickel syndrome, affecting glucose transport in the brain and other tissues. GLUT1 deficiency syndrome is a mutation in SLC2A1 gene, which encodes GLUT1 and can lead to GLUT1 deficiency syndrome, a rare neurological disorder. It results in impaired glucose transport in the brain, leading to development delays, movement disorders, seizures, and other neurological symptoms. Cancer. GLUT1 is often overexpressed in cancer cells, contributing to their increased glucose uptake and metabolism. Known as a Warburg effect, targeting GLUT1 is being explored as a potential therapeutic strategy in cancer treatment. The end. Hope it will help you all. Leave a like and subscribe. Thank you for watching.